Hello, everyone. My name is Chheda, and today I'll be talking about my project, the Periodontal Ligament Regeneration, with CRISPR activation and pluripotent stem cells. So, to get started, the essential issue that we wanted to solve was in periodontal disease, which impacts over 1 billion people worldwide. One of the most common side effects is the degeneration of the periodontal ligament. Now, we wanted to challenge the current limitations in single gene therapy, where we wanted to use CRISPR activation to upregulate two genes simultaneously, which are scleraxis and mohawk, both of which play important factors in regenerating the periodontal ligament. Now, as you can see on the image on the left, our CRISPR activation model essentially has a set of items, the dCas9 molecule, the V, P, and the R transcription factors, as well as the guides of both of our genes of interest to essentially upregulate both of these genes of interest simultaneously. Now, we in the CRISPR activation model first wanted to build our toolkit, which also included valproic acid, an epigenetic modulator, which helps loosen the chromatin to help it become more accessible so that we're able to upregulate these genes easier. Now, the most important part was the usage of the human pluripotent stem cells, which in the future have a direct translation to stem cells in the periodontal ligament. Now, as you can see from our results, some of the key findings that we had was that Mohawk on its own um, showed a 10-fold increase, whereas with valproic acid showed over a 38-fold increase. And then Mohawks in the multiplex format, which is Mohawks and Sclaxis put together, increased from 4-fold to over 18-fold with the addition of VPA. Now, Scleraxis went from 200-fold to over 500-fold alone, and then in the multiplex format went from around 180-fold increase to a 400-fold increase, both cases showing that we were able to effectively multiplex and upregulate both of these genes of interest using the CRISPR activation model in human iPSC cells. So now in the future, what we hope to do is integrate a third gene of interest, which is CTGF, essentially another key factor in the periodontal ligament that helps with connective tissue repair. Secondly, we also want to see how VPA is really controlling each of these genes of interest. We know that it loosens the chromatin, but we want to see specifically what the mechanism of it is. And lastly, and most importantly, is translating our CRISPR activation work to the in vivo rat model to really see the usage of lipid nanoparticles and how we're able to translate this research into a clinical study. Thank you so much. And I would like to thank Albert Wang, Dr. Christopher Recupero, and Dr. Cheng Li. Thank you so much.